Hello everyone, welcome back. So this is week 6 of the NPTEL course Fundamentals of Protein Chemistry. This course is coordinated by Professor Das Gupta and from the Department of Chemistry, IIT Kharagpur, and I am Bias Mukherjee. I am a research scholar from the Department of Bioscience and Biotechnology, and in today's class we will discuss a few topics of enzymes and we will solve corresponding questions and answers, especially MCQs, MSQs and numerical aptitude questions. Okay, so let's begin with today's class. So to give you a small overview of what is there in today's class, we have enzymes, we have different activity of enzymes and their regulation, and we also have the different types of enzymes. Then we are going to talk about proteins and proteases and the sites at which proteases cleave. Now all of these are very important topics for your exams. So let's start with today's class. Okay, right, so we will start with the basics. Now let's talk about enzymes. As you know that enzymes are biological molecules which catalyze biochemical reactions with very high specificity. Now the majority of enzymes are made up of proteins, but enzymes can also be made up of RNA molecules, and these enzymes are called ribozymes. Okay. Now enzymes accelerate chemical reaction by lowering the activation energy. Now you guys have already learnt about enzymes possibly in school or in college, right? So we will just brush upon these concepts quickly and then we'll get into the parts that will be new to you. Okay, so and this is how enzyme influence a chemical reaction, and enzyme activity can be regulated through various mechanisms including allosteric regulation. Now in our last class we were talking about allosteric regulation. Then there can be covalent modification in the enzyme which influences its structure, folding and function. Okay, and then there can be feedback inhibition. So in feedback inhibition, what happens is when the product, let's say that is produced in a very large amount, the product inhibits the activity of the enzyme, ensuring that only a specific level of the product can be produced at a time, okay, depending upon the need of the cell. Now, enzymes play essential roles in almost all biochemical processes, from metabolism, digestion, DNA replication to cellular signaling. So insights into the enzyme function provide opportunities for the development of novel therapeutic strategies, diagnostic tools and biotechnological applications. Now in this figure you see that this small substrate molecule, it is binding to the enzyme, and this is how the enzyme has a pocket for substrate binding, and the substrate is kind of complementary to it and it comes and binds to it. Now you know that there can be lock and key model and there can be induced fit model. So we will talk about all of that very soon. Okay. Right. So now for understanding enzymes, there are some terms which you need to brush upon. Okay, the first one is coenzyme. Now, what is a coenzyme? Coenzyme is an organic molecule. Now, there can be questions related to these terms. That is why it's important to just revise. Okay, so coenzymes are organic molecules often derived from vitamins that assist enzymes in catalyzing reactions between different chemical groups. Okay, by carrying chemical groups between enzymes and their substrates. Now, what is a cofactor? Now, cofactors are inorganic ions or non-protein organic molecules that are required by for the proper functioning of the enzymes. Now, that is the difference between a coenzyme and a cofactor. So, remember that. Next thing is apoenzyme. What is an apoenzyme? So, apoenzyme is something which is not functional yet. The enzyme that is not functional yet because it does not have its partner with which it can bind and then get activated for its catalytic activity. Okay, so the protein portion of the enzyme that lacks its cofactor or coenzyme and thus is inactive, that is the apoenzyme. The next one is holoenzyme. So hollow means full. So apoenzyme plus the cofactor or coenzyme together makes up the fully functional enzyme, that is the holoenzyme. Okay, now we'll talk about the active site of an enzyme. So what happens in the active site? So active site has certain very important characteristics. Okay. For example, specificity of the active site, that is one of its very special characteristics. So it is having complementary shape to the substrate with which it is going to bind. Now it may or may not have complementary shape because the substrate can undergo conformational change and then bind to the enzyme ultimately, okay, based on the shape of the binding pocket of the enzyme. Next thing is the catalytic residues. Now catalytic residues will have chemical groups on them which will be oppositely charged compared to the substrate, so that binding is feasible. Then induced fit. So what happens in induced fit? You guys know. Let's say this is the shape of the substrate and this is the binding pocket of the enzyme. So when it comes close by, what happens? It changes its shape and then it binds. It becomes complementary and then it binds. Okay. So unfolded protein has no activity, but only when the protein is in its tertiary structure, folded structure, the binding pocket gets arranged in such a way that all these binding residues, they come very close by and they are forming this shape which is complementary to the shape of the substrate. Okay. Now active site of an enzyme is a specific region or pocket on the enzyme substrate where the substrate binds and undergoes a chemical reaction. It is typically a groove or a cleft formed by the arrangement of amino acid residues in the enzyme's three-dimensional structure. Now specific orientation, chemical group and charge of the residues. Now these are the three main important properties of the active site that make up the catalytic center for binding to the specific substrate during the enzyme catalyzed reaction. Okay, right. So let's talk about the enzyme substrate formation. So see, how does an enzyme function? The enzyme is there, free enzyme, then it binds to the substrate and forms the enzyme substrate complex. Then what happens? The substrate is converted into the product and enzyme product complex is there, and then the product is freed and the enzyme is once again free, and it repeats this cycle. So now the rate at which the enzyme substrate complex is formed, that is denoted by K1. The rate constant for the reverse reaction, that is enzyme substrate to enzyme plus substrate. So it is basically breaking up. That is K minus 1. And then enzyme substrate to enzyme product formation or enzyme plus product formation, this is K2. So K2 usually does not have a reversible reaction. So once the substrate is converted into the product, it is not converted into the substrate back once again. Okay. Right. So now the enzymes, they are divided into 6 different groups. Now they are classified into 6 different groups. What are they? If they are involved in oxidation and reduction reactions, they are called oxidoreductases. The second group is transferase. That means the enzyme is responsible for transferring a chemical group to another chemical moiety. We are transferring of functional groups. Then there is hydrolases. So hydrolases means it is catalyzing a hydrolysis reaction. That means a reaction where something is breaking up, that is hydrolysis. Okay, and water is released in that reaction. Then there is lyases. So lyases involve breaking of double bonds, so elimination reactions to form double bonds. So it is participating in elimination reactions to form double bonds. And then there is isomerase. So from this compound, it is becoming this compound. Both of these are isomers, and isomerase is participating in this reaction. So just a rearrangement of the bonds are happening. The other group is ligases. So ligase is joining of two moieties and using ATP. Okay, so these are the 6 groups that enzymes can be divided into. Now we'll start with our questions and answers. Okay, so the first question for you guys is, the enzyme active site should preferably be more analogous to, one is the product, second is the substrate, and third is the transition state. Now to give you a hint, we have an image here. So based on this image, you tell me. So you have to keep in mind the lock and key mechanism and the induced fit mechanism. Okay, you have to keep both of these in mind. So which one do you think would be the right answer? So from the reactant, the reactant, the substrate is converted into the, so enzyme and substrate together forms the transition state, and from there the intermediate state is formed, and from there the product is formed, right? Okay, so we look at the answer. Okay, so you're saying that it is A. Very good. Let's look at the answer. No, the answer is actually B. So B is the transition state. So now the transition state should be complementary to the enzyme's active site. Okay, because the substrate can undergo induced fit mechanism and change into the required shape, and then the transition state has to be more analogous to the enzyme's active site. Right. So now, what is the difference between this intermediate state and transition state? So that is something that we have to understand, right? So see, there can be more than one transition states, but there is often only one intermediate. So intermediates are more stable states, and in reactions with intermediates, with one intermediate, two transition states are involved. The slowest step or the rate determining step has the highest activation energy barrier, and the formation of the intermediate is actually the slowest step. Okay, this is the rate determining step. Now it is very difficult to capture transition state, but then it is still relatively easier to capture an intermediate state in a reaction. Okay, so now activation energy, the energy required to reach the transition state from the reactant, that is the ground state. So ground state to transition state is actually the activation energy, and the enzyme's activity is to lower this activation energy to make the transition state reachable, more reachable. Okay, so AE barrier must be exceeded for a reaction to proceed, and lower activation energy barrier, more stable the transition state, because this is the energy axis. More the energy, less the stability, right? So now the higher transition state concentration, more likely the reaction is to proceed in the forward direction. Okay, right. So once again, here the enzymes, the different classes of enzymes have been explained for you. So you can go through this, but then we have already discussed this. So I will skip this slide and you can start answering this particular question. So we have a few questions now, and then we'll move on to other topics. Okay. So here there is a compound, and this compound gets changed into another compound. So see what is happening. This single bond is changing into a double bond, right? So this hydrogen is released and double bond formation is taking place in this case. So what is happening? Which type of enzyme is more likely to be involved in this reaction? Now we have read about this. Try to understand what is going on over here. C. Okay, let's see. Right, answer is ligase. Ligase is the one that joins two chemical moieties together using ATP. That is ligase. But ligase is something that makes sure that a double bond is formed between two chemical groups. Okay, so the right answer is ligase. Okay, now we have the next one. So see what is happening here. Which group of enzymes would be involved in this reaction? Shown that involves the conversion of carbamoyl phosphate to carbamoyl aspartate. So some kind of reaction is happening. Which of these enzymes would be involved? What do you think? The first one is hydrolase, second is ligase, the third one is oxidoreductase. So some kind of transfer is going on. So which enzyme do you think is involved? I am giving you too many hints now. Should be able to answer. Chalo, it's a simple question. Should be able to answer this. C. C is oxidoreductase. What is wrong? So the right answer is D. Why is that D? because see, a transfer is happening. So if we go back to this slide, you will see that 6 different groups of enzymes are there and their functions are mentioned there. Right, so transferase is responsible for transferring a particular group from one chemical moiety to another. Now here in this reaction, what was happening? There was this transfer of this particular chemical group, right? So this, so this aspartate group was getting transferred to this carbamoyl group, and that is why it's a reaction performed by transferases. So we'll go for the next question. So next, there are 2 questions for you to answer. The first question says the enzyme part which associates, now enzyme part which associates with a non-protein part to form a functional form of enzyme. What is it called? Non-protein. And the second question says methionine synthase catalyzes the conversion of homocysteine, which is an amino acid, into methionine in the presence of vitamin B12. So here vitamin B12 plays the role of A or N. Okay. So vitamin B12 is an organic moiety. So then what should it be? Is it an apoenzyme, holoenzyme, cofactor, prosthetic group? And for the first question, it's a non-protein part. So that is why we discussed that, because it's very important that you remember the difference, okay, between a non-protein part and a organic moiety helping in catalysis. A. Okay. A is for which one? Second question or first question? First one. Okay. Okay. First one. So you're saying that it is A. What about the second one? So if we go back to this particular slide. Okay, so we have come back to this slide. So see, non-protein organic molecule, inorganic ions are non-protein organic molecules that are required for proper functioning of enzymes. These are cofactors. And coenzymes are organic molecules that are derived from vitamins. So then for this particular question, the right answer for question number 1 is C, that is apoenzyme. So the enzyme part of the enzyme which associates with non-protein part. So the non-protein part is your cofactor, right? So it can be cofactor, it can also be coenzyme for this case, because it is not mentioned whether it is organic or inorganic, just non-protein, right? So apoenzyme plus the cofactor or coenzyme gives you holoenzyme. But in the second case, it is mentioned that this is a vitamin B12. So normally, if it's a vitamin, then it should be a prosthetic group or coenzyme. But then you can read about this. You can find out in this case whether this vitamin B12 is playing the role of a cofactor or coenzyme. Okay, because the answer given to us was cofactor. So you can just double check if this is a cofactor or coenzyme. Okay. Now we come to the properties of enzyme catalysis. So enzymes increase the rate of reaction by lowering the activation energy required for the reaction to happen, thereby speeding up the process that is essential for life. Okay. So the enzyme and substrate together, they come together, and the activity and activation energy is lowered. So the transition state is reached quicker and then intermediate state is reached if there is any, and then the enzyme product complex is formed and then it splits up into enzyme plus product. So that is how the reaction proceeds. Okay, now you already know that enzymes are very sensitive to temperature and pH change, right? So enzymes perform best at a specific temperature range or specific pH range. This small range is called optimal temperature or optimal pH. Okay, so enzyme requires an optimum temperature and pH for their action. The temperature or pH at which the compound shows its maximum activity is called optimal temperature or pH respectively, and the temperature or pH more than the optimal may alter the molecular structure of the pH of the enzyme. So let's say that the enzyme is kept at a temperature that is extremely high. Then what happens? The protein gets denatured. Right? It's after all a protein. And if it is kept at a pH of, let's say, very low or very high compared to its optimal activity, then what happens? It can get precipitated. Right? So the general normal optimal range is between 5 and 7, pH of 5 and 7, and for temperature it is different for different enzyme. Okay, now heat. The increased energy level of the molecule disrupts the bonds in the enzymes and between enzymes and substrate. That is, it can be hydrogen bonds and ionic bonds. Hence, denaturation results in loss of the 3D structure or functional native structure of the enzyme. If it is kept in too much cold temperature, then what happens is the molecules are not functioning, they are moving very slowly, and that is why the chances of collision between the enzyme and the substrate decreases. As a result, the activity of the enzyme also decreases. Right, so we have graphs, we have curves for all the different parameters, be it temperature, be it pH, be it enzyme concentration, be it substrate concentration. Now these graphs you have to remember. Okay, graphs you have to remember. So big influence. So see, temperature is a big influence. More heat means more kinetic energy in the molecules, more collision and more chances of your enzyme activity. But if it is too high, then it gets denatured. So if the temperature is more, then very rapid decline happens. For pH, it is best at an optimal range. Okay, so if it is higher or lower, H plus ion concentration is more or less, it interferes with the enzyme structure. Next, the enzyme concentrations. Initially, if you increase the enzyme concentration, it will increase, the activity will increase. But once the enzyme concentration has reached a particular level where your substrate has been utilized, then what will happen? It will become constant, right? Because all of the enzymes are bound to all of the substrate molecules. More substrate needs to be added, only then it will further increase, right? What about substrate concentration? Exactly same. So initially when you are adding, it is increasing, then it becomes saturated because the number of enzyme molecules is now all occupied. Okay, now enzyme catalysis. Now enzymes are extremely specific. Now they are so specific, you will be surprised. We'll talk about that right now. So see, enzymes are very specific as they bind to substrates with distinct chemical groups only in certain orientations. Now enzymes can even identify whether a particular atom is present in the cis conformation or trans conformation of a particular bond. Now they are very specific. Now not all enzymes are specific. Right, some enzymes are specific. Some enzymes can act on substrates of a particular group also. So we'll talk about that. So see, absolute specificity. So this means that the enzymes will catalyze only one reaction. Then there is group specificity. So the enzymes will catalyze, will act on molecules that have a specific functional group such as aminophosphate or methyl groups. Group specificity, chemical group specific. Next is linkage specificity. The enzyme will act on a particular type of chemical bond regardless of the rest of the molecular structure. It only cleaves, let's say, one particular bond. Okay. Then there is stereochemical specificity. That means the enzyme will act on only one particular steric or optical isomer. Okay. Now see, specificity of enzymes can be bond specificity. group specificity, substrate specificity, optical specificity, geometric specificity, and cofactor specificity. So see, for example, alpha amylase, this one, the example is of stereo specificity. Okay, so what is happening? It will only act on alpha 1,4 linked glucose based on the orientation of this particular bond. It's between particular carbons in the two glucose molecules that are joined together. So that is the alpha 1,4 linkage. Now, if there is a beta 1,4 linkage, then what happens is alpha amylase will not act on it anymore. So alpha amylase is only specific to alpha 1,4 linkage. So that is an example of stereo specificity. Okay. So this is between starch and cellulose. Now we come to a question. Okay. So they are asking you to choose the incorrect statement. Okay. So the first statement says that the free energy of the intermediate, the free energy of the intermediate must be greater than that of the transition state. And then the transition state with the highest energy always corresponds to the rate determining step. The free energy of the intermediate may be greater than that of the reactant. By catalyzing the reaction, the enzyme must decrease the free energy of the intermediate. So you have already read about intermediates and transition states. So to give you a small clue, we have the figure here. Okay, so you can refer to this figure, and from here you can tell me whether the free energy of the transition state or the intermediate state should be greater, and whether it should be more than the reactants, etc. Okay, so based on this figure, which one do you think is incorrect? Which of the statements are incorrect? Okay, and this is a multi-step reaction. Okay, so we can look at the answer. So see, the answer is number D. That is 1 and 4 are incorrect. So the free energy of the intermediate must be greater than that of the transition state. That is not correct. You can see from the figure that the free energy is in fact lesser than that of the transition state. Why? Because intermediate is more stable. It is more spontaneous. If the free energy would be more than that of the transition state, then it would never go to the intermediate. Right? It would never go. Now, the fourth one says, by catalyzing the reaction, the enzyme must decrease the free energy of the intermediate. So intermediate is actually, the enzyme's activity is to lower the activation energy. Activation energy is the energy gap between that of the transition state and the reactant. It does not concern the intermediate. Right? So if we go back to our previous slide, we will see that it is very clearly mentioned that the activation energy is the energy between the transition state and the substrate, right? So it has nothing to do with the intermediate here. That is why it's very important that you follow whatever we have learned until now, and then it will be easier for you to answer these questions. Okay, so we go to the next question. So the next question should be simpler for you. You have already seen this graph in the previous slides, right? So the graph shows the effect of what factor on enzyme activity. For two factors, this is the graph that we get. Okay, so which are those two factors? One of them is mentioned here and the other one is also there, which you guys know. Try to answer. Is it temperature? Is it substrate concentration? Is it pH? Or is it activation energy? B is substrate concentration. That is correct. So for substrate concentration and for enzyme concentration, This is the graph. But then for temperature and pH, you know that it is a bell-shaped curve, right? Okay. We go to the next question. Now, the next question is something that you have to remember. So, in our last class, we were talking about turnover number and specific activity. Now, I had told you that in today's class, we will be discussing all of this, right? So, since this is enzymes class dedicated specifically for enzymes, we have to discuss this. And this is very, very important because you get a lot of questions from these topics. Okay. So now for your numericals, there will be these relations between classical units and new international units. So one of these will be given, one of the values will be given, and you will be asked to find out the other. So you need to know which one is related to which one and which one means what. Okay. So first thing is turnover number or molecular activity. So this tells us how many maximum molecules of substrate can be converted into product per catalytic site of a given concentration of enzyme per unit time. Okay, so that is kcat. So turnover number or molecular activity is denoted by kcat, and that is Vmax by enzyme concentration. That is enzyme total. ET is enzyme total. Okay, so the units of turnover number are moles of product per second divided by moles of enzyme or second inverse. This is its unit, second inverse, right? So the number of molecules of the substrate transformed to product by enzyme per unit time is defined as enzyme activity. You have to remember the differences, okay? And specific activity means the activity of the enzyme per milligram of total protein. Total protein means total enzyme, okay? So now the classical units are micromole of, okay, so what is the unit of enzyme activity? That is micromole of substrate transformed per minute. That is the enzyme activity, and it is written in unit. Okay. What about specific activity? This is micromole of substrate converted per minute per mg of enzyme. This is there. And the unit is unit per milligram enzyme. Okay, now what about molecular activity? So molecular activity is micromole of substrate converted per minute per micromole of enzyme. That is your molecular activity or turnover number. Okay, and the unit is unit per micromole enzyme. Now for international unit, now you have to understand the relation between these two, classical and international. Okay, so for international unit, the unit of enzyme activity is moles of substrate divided by second. So that is moles of substrate converted per second. It is shown in cat. Then there is specific activity, which is moles of substrate converted per second per kg of enzyme. That is cat per kg enzyme. Then there is molecular activity, which is moles of substrate converted per second per mole of enzyme. So that is cat per mole of enzyme. Now it sounds confusing, I know, but then with practice you will be able to understand all of these and remember all of these, because for your numericals you have to. Okay, you have to remember all of these. Okay, so now we have a question here. So let's try to solve this. Okay, so 10 microgram of carbonic anhydrase and the molecular weight is given to you in the presence of excess substrate exhibits a reaction rate of this micromole per minute. So at substrate concentration, this molar, what is the, and the rate is this particular micromoles per minute. So now they're asking you what is Vmax, what is Km, and what is Km, by the way? Michaelis-Menten, right? Michaelis-Menten constant. And what is K2? So K2 or Kcat, these are synonymous, okay? Right, so we will look at how to solve this question. So see, if this kind of question comes, then this is how you will solve it. So the rate in presence of excess substrate is Vmax. So from your Michaelis-Menten equation, you will get this, and Vmax is equal to this amount micromoles per minute. Now, at substrate concentration, this, the rate is given as this, right? Which is half of Vmax. Therefore, if it is half of Vmax, then you know from the Michaelis-Menten equation you can calculate Km, right, which is this constant, and the value of the constant comes to be 0.012 molar. Now, you know by definition that the Km is equal to the velocity of conversion of substrate to product at half of substrate composition, right? Half substrate concentration. So that is your Km. Next, this may also be determined using the Michaelis-Menten equation. And then divide Vmax by micromoles of total enzyme to find the Kcat, and that will come to this particular value. So this is how you will have to go about with all the questions. Okay. Now we have one more question. So this is also a numerical. Let's see. How much does an enzyme reduce the activation energy of a reaction at 25 degrees centigrade for the reaction to occur 70 times faster than in the absence of the enzyme? Okay. So for solving this kind of question where only temperature is given and the rate of the enzyme's function that is given. So for that you will have to use this particular equation. This is called Arrhenius equation. So here what are the things that are going to change? First, the rate of the reaction is going to change. Rate of reaction is K, right? A is Arrhenius constant. Activation energy, you see how much does an enzyme reduce the activation energy. So the change in the activation energy is something that you have to find out. Now R is a constant. T they have even mentioned. So here the T should be in Kelvin, right? So this is how you have to find out. So from if you are trying to solve this question, then this is how you have to solve it. Okay, so the right answer is D. Now you can practice at home. Okay, with taking your time you can do it. Here I'll just tell you how to do it, and then later on you can sit down and you can solve properly and do. Okay, so K of enzyme divided by K of no enzyme. It is 70, right? Because it is decreasing by 70 times in absence of enzyme. And what else is going to change? This activation energy is also going to change, right? So from this Arrhenius equation, you can see that with change in temperature, with increase in temperature, the rate constant increases in a logarithmic scale. So based on that, you can use this particular equation, and that is ln of rate constant gives you minus activation energy divided by R into 1 by T plus ln of A. Now A is a constant, so for this equation you do not need this, but this is how you can actually solve this question. Okay, now we go to the next questions. There are two questions here. Okay, so the first question says the catalytic activity of the enzyme. So you have to choose the correct statement from here. Okay, so the catalytic activity of the enzyme depends on the native protein composition. All enzymes must be proteins. Enzymes exhibit highest efficiency at 25 degrees centigrade, and all enzymes are stereoselective in nature. Now, very clearly we know that not all of these sentences are true. Correct. So you have to tell me which one is a correct statement. For your second question, you have to choose the incorrect statement about an enzyme. Okay, so compared to an uncatalyzed reaction, an enzyme speeds up the rate at which a chemical reaction approaches equilibrium. Enzymes make a reaction thermodynamically favorable. Enzyme decreases the delta G of reaction as well as the delta hash. Now we will see what this is later on. Okay. In the presence of an enzyme, all thermodynamic parameters of a reaction are altered. So now you are going to tell me which of the statements in the first question are correct and which of the statements in the second question are incorrect. Okay. You can answer in the chat box. So the first one is easy. First one you should be able to do. so first one is A. That is correct. Yes. So for the first one, it is A. And for the second one also, the answer is actually A. Now, why is that so? Because compared to an uncatalyzed reaction, the enzyme speeds up the rate at which, second one is also A. That is correct. Yes. So, A tells you that the enzyme speeds up the rate at which a chemical reaction approaches equilibrium. So, that is not correct. It just reduces the activation energy and it pushes the reaction towards completion, that is formation of the product, not equilibrium. Okay. Now, the third, okay, sorry, one second. Okay, sorry. So the first one is actually correct. The first statement is correct. So it actually speeds up the rate at which the chemical reaction approaches equilibrium. And the second one, see, enzyme makes the reaction thermodynamically favorable. That is not correct. The enzyme is not so powerful that it can do that. Okay. And the next one, see, the enzyme decreases the delta G of the reaction, which is not true, and as well as delta G hash. So we will look at what is delta G and what is delta G hash very soon in the upcoming slide. And in the presence of enzyme, all thermodynamic parameters are altered. That is also not correct. Okay. Right. So for the first question, now this one was simple because we know that not all enzymes are proteins. Some enzymes are ribozymes. Some enzymes such as ribozymes, these are made up of RNA molecules. Right. The next one is enzymes exhibit highest efficiency at 25. No, that is a very generalized statement because different enzymes are active at different temperature. For example, if you look at bacterial enzymes, they are more active at around 37 degrees centigrade or so. Okay. Now, all enzymes are stereoselective in nature. That is also wrong because enzymes have different kinds of selectivity and that is there in this presentation. Right. So, you can just go back and have a look. Okay. Now, we come to this delta G and delta G hash. So, delta G is actually gives free energy and delta G hash is the activation energy. Enzyme decreases the activation energy. Okay. And there is something which is called the Eyring's equation. So, this gives the like this comes from the transition state theory, and we have learnt about transition state, and it links the activation energy with rate constant. Okay, so this is an important equation. It's very important that you remember this. So there can be numericals from this equation as well. Okay. So now if the delta G hash is lowered, that means the transition state can be achieved quicker and the reaction can happen faster. And if the delta G is more negative, that means the reaction is spontaneous and it will happen. Otherwise, it will not happen. Okay. Now we come to something that can be new to you people. Okay. So these are proteases. Now that we are talking about enzymes, enzymes have different functions, right? So one of their functions is to cleave proteins or peptides. Okay, so there are different proteases. Now these can be exopeptidases, which means that they are cleaving the last residue only. So this can be N-terminal and C-terminal. They are cleaving only the last residue. They can be endopeptidases. So that means they are cleaving in between. There can be dipeptide peptidases, tripeptidyl peptidases, etc. Right, so there are different types of peptidases and they have different specificities. Now for your exams, we will talk more about serine proteases and we will talk a lot about the cleavage site of different proteases because this is a question that gets repeated in every exam. Okay, so now proteases, these can be peptidases, so that is they are acting on peptides, or they can be proteinases, so they are acting on big proteins. Okay, are enzymes that break down proteins into smaller polypeptides or single amino acids. Now proteases are found in all living organisms and they help in protein degradation, programmed cell death, protein activation and pro-protein extracellular matrix remodeling, cellular signaling, etc. Now you know that there are some proteins which need to be cleaved, only then they will become active, right? And then there are some proteins which undergo some kind of, let's say, cleavage. So there are these zymogens. Zymogens are enzymes that undergo cleavage. Only then they are going to be active enzymes. Okay, so these are different kinds of proteins that require cleavage for either cell signaling or other different purposes. The proteases are classified based on their catalytic residues such as serine, cysteine, aspartic acid, glutamic acid, and metalloproteases. So these are the different important residues in proteases that are necessary for their function. Now we will be talking about serine proteases. So in serine proteases, there are three important residues. One is a serine residue, one is a histidine residue, and then one is an aspartic acid residue. Now these three form a catalytic triad in the binding pocket. So catalytic triad means that there is some kind of interaction going on between these. Okay. So now the arrangement is such that it polarizes the serine hydroxyl group, and the histidine becomes a proton donor, and stabilization of this is done by aspartic acid. Now see, serine, in case of this particular serine protease that we are talking about, serine 195 in the catalytic triad acts as a nucleophile in trypsin and chymotrypsin. This serine is located in the conserved region of the enzyme's active site and plays a crucial role in the hydrolysis of peptide bonds. So it is cleaving peptide bonds, right, of the protein. Now histidine, this functions as a general base. So that means it abstracts a proton from the serine's hydroxyl group, enhancing the nucleophilicity of the serine residue. This action helps in the formation of a highly reactive alkaloid ion, the one that you see in the right side figure. That alkaloid ion is formed and it is highly nucleophilic. Aspartate residue helps in stabilizing the positive charge that develops on the histidine during this catalytic process. It helps in maintaining the proper orientation and ionization state of the histidine. Okay. Now we will go to the entire process. So now this has been taken from Leninger's biochemistry. So you can go there and you can go through this entire process, how this substrate is undergoing cleavage because of this catalytic triad in the enzymes. Okay. So you can go through it. Now we will talk about this particular enzyme. Okay. There's a question, and in this question they are asking you about trypsin. So what will likely affect mutating aspartate residue in the catalytic triad of trypsin to asparagine? Now what is the difference between aspartate and asparagine? Now aspartic acid has this side chain carboxylic group, right? But asparagine has a CONH2 group. So this OH is now changed to NH2 group. So how is it going to affect the activity of this trypsin? Okay, so turnover number will be increased. Catalytic efficiency will be increased. Catalytic efficiency will be unchanged and turnover number will be unchanged. So these are the kind of questions for which you need to know the difference between turnover number and catalytic efficiency. Okay, so try to answer this. Okay, so you can try to answer this question. Okay, now we can look at the answer now. Okay, so the right answer is catalytic efficiency will be unchanged. Now you can go back to the definition of turnover number and catalytic efficiency, and from there you can understand how that is happened. Okay, now we will move on to another question. So this is a huge question. Now let's try to understand what the question says. Okay, so in chymotrypsin protease is a digestive enzyme component found in pancreatic juice. It is active in the duodenum, where it performs proteolysis and breakdown of proteins and polypeptides. Now, in the above mentioned protein, there is this histidine residue which is present in the catalytic triad. This interacts with this serine residue to increase its nucleophilicity in the latter, right? It forms this alkaloid ion. Now, on the other hand, this particular aspartate residue plays two roles in the catalytic process. One is to stabilize the positive charge on the adjacent histidine residue by a strong electrostatic effect, and the other is to fix the alignment of the catalytic residues around the active site. Now, histidine 57 enhances the nucleophilicity of serine by acting as one is acid catalyst, base catalyst, proton donor and proton acceptor. Now you have to choose the right options, okay? Whether it's acting as a base, whether it is acting as a proton donor, proton acceptor, what is going on. Okay, so two answers will go hand in hand. Acid catalyst, if you understand the meaning, you will be able to answer whether it's a proton donor or proton acceptor. Right, so that is how you will be able to solve it. You can try answering this question in the chat box. So histidine is going to act as what? Base catalyst and proton acceptor. So if we go back to what we studied here, see, histidine residue is performing as a general base and it is taking away a proton from the serine's hydroxyl group, right? So acid means acid's property is to release hydrogen, right? Only then it is an acid. But then a base is supposed to accept hydrogen. So if it is acting as a base, it would definitely be a proton acceptor. Correct? So the right answer in this case is B. That is number 2 and number 3. Now we go to something very important. Okay. Proteolytic enzymes. So just like chymotrypsin that we talked about, there are so many different common endopeptidases, and these are very important because see, trypsin. Now trypsin has a specificity where the C-terminal side of the positively charged residues, it cleaves. So it cleaves when there is RK there. Okay. So arginine and lysine, and it is going to cleave on the C-terminal side of arginine and lysine only if there is no proline present. So here in these questions, they will give you a sequence of a peptide and they'll ask you, if we are adding, let's say, trypsin, how many fragments are you going to get? Or how many places is it going to cut? So these are the kind of questions that you're going to get. Okay. So now then there is chymotrypsin. Chymotrypsin cleaves at WYF, that is phenylalanine, tryptophan and trypsin. Okay. And there should not be any proline on the right hand side. Next thing is elastase. So what happens for elastase? It cuts at GASB and there should not be proline. Okay. Pepsin. Pepsin cleaves at the N terminal. Now try to understand the difference. So N terminal of these different residues and there should not be any proline at the N terminal. Next, for thermolysine, it cleaves at the N terminal of any hydrophobic amino acid excluding tryptophan. Okay, and R-1. That means the residue on the N terminal should not be proline. Okay, so this is how you have to remember where which enzyme cuts. Now, we talked about zymogens, right? There is an even longer list for you waiting at the end of this PPT. So in that list, there are many chemical agents, there are endopeptidases, there are exopeptidases whose cleavage site you will have to remember because those are very important for exams. Okay. Now, so see, there is this zymogen. Now zymogen undergoes a kind of cleavage and then it becomes functionally active. Okay, so proteolytic enzymes are biosynthesized as larger inactive precursor known as zymogen. Why are zymogens not in functional form? Why? Zymogens do not consist of active sites. Zymogens have more steric crowding than the active enzyme, so it cannot bind to the substrate properly. Zymogen does not contain the same amino acid sequences as the active form. Zymogen have a different conformation than the active form, so the active site becomes distorted. So what do you think is happening in case of a zymogen? Make a guess, or if you can figure out the right answer, you can just type in the chat box. Okay, it shouldn't be very difficult. Try to understand. Try to think what can be happening. So based on the figure, what do you think is happening? Chalo, think about it. What is happening? So we look at the right answer. Okay, so the right answer is D. Now zymogen is having a conformation of the active site that is different from the functionally active site. That is why it is not able to bind to the substrate and it is not able to catalyze it. Okay. Right. Now we come to something that you will have to remember. So there are these N-terminal proteases that we talked about, like thermolysine, right? So there are more. For example, aminopeptidases. And then there are ASPN and glutamic N. So these are different residues that cleave. Then there are C-terminal proteases like trypsin that we talked about, chymotrypsin, elastase, and three of these are very important. See, carboxypeptidase A, B, and C. Carboxypeptidase A removes the C-terminal residues except proline, lysine, and arginine. Carboxypeptide B removes only C-terminal, arginine, and lysine. And carboxypeptidase C does not care about any amino acid. It just removes all C-terminal residues. Okay. Now we come to chemical cleavage. So besides these enzymes, there are also certain chemicals that are used for cleaving peptides and proteins. Okay. For example, cyanogen bromide, CNBR. This cleaves at the C terminal after methionine. Then there is hydroxylamine. This cleaves between asparagine and glycine, okay, at the N-terminal side of the glycine. Then there is Edman degradation. So Edman degradation is a sequencing technique where it sequentially cleaves N-terminal residue for sequencing, okay. And for this, the compound that is used is PITC. So in the next slide, Edman degradation is explained in detail, so you can go through it. This was also taken from Dellinger's biochemistry, so you can refer to it for better knowledge. Okay. Now proline often blocks cleavage because its rigid cyclic structure prevents proper recognition or bond distortion. Then at the C-terminal side, proline blocks most endopeptidases such as trypsin, chymotrypsin, elastase, and lysine. Next we have at the N-terminal position, proline strongly interferes with Edman degradation and aminopeptidases. Then carboxypeptidase A to C are exopeptidases. So that means they are cleaving the exact N-terminal residues. Cleaves one residue at a time from the C terminus with different specificities. Right? Okay. So in this slide, you have the entire Edman degradation explained in details. we come to the next few questions. So the first question says a rate versus pH curve with an inflection point at pH around 4. So optimal activity is at pH 4. Suggests the involvement of a dash in the catalytic step. So hydrophobic amino acid, acidic amino acid, basic amino acid and metal ion. So inflection point of pH 4 suggests involvement of dash in the catalytic step. Right. Next we have the next question says this particular sequence is given to you. Where do you think elastase is going to cleave? First one is B. Alright. Good. Try to answer the next one. Now elastase cleaves at GAS and B. Okay, GASB. It cleaves on the C terminal of GASB, ensuring that there is no proline next to it. Right, so you're trying to answer this one. We'll look at the answers. So see, yes, so the first ones, yeah, that, okay. So it's actually five different fragments will be formed, okay. Why? Because there are many cleavage sites. There are actually 5 cleavage sites, but for one, the cleavage will not be possible because of the presence of a proline on the C terminal. But for the other 4, it will be possible, and that is why 4 different cleavages will be happening and 5 different parts will be there. But if you look at this particular V, this V might not get cleaved because this elastase is it's an endopeptidase. It is not an exopeptidase. Okay. So the answer says that there will be 5 different cleavage, 4 different cleavages. So that means 5 different fragments will be formed. Okay. Now, for the next class, there will be a lot more questions on enzyme inhibition discussed with you people. Okay, so we will leave that for the next class. So for the next class, we have this enzyme regulation, we have entire enzyme kinetics and inhibition, and we will discuss all of that in the next class. So thank you so much for your time, and we can end the class today. If you don't have any more questions, we can actually end this class. Thank you so much for joining.